To continue with our discussions, we go now to carbohydrates, one of the most important medium that is used for the growth and pigmentation of bacteria. And of course, when you say carbohydrate, this refers to sugars. It contains a polysaccharide also. And carbohydrates, no, used to determine whether the bacteria is produce acid or base and gas from the bacteria itself. Okay, the concentration, take note, is 0.5 to 1% only. Nutrient agar, so this one is used for the non-fastidious bacteria. Mga non-fastidious bacteria, these are bacteria that do not require special nutrient, while mga fastidious, they require special nutrient. So mga non-fastidious, wala na. Okay, a common agar like nutrient agar is one of the common or basic agar that is used, so that bacteria can survive, can grow in that certain common agar only, kasi they are known to be non-fastidious. Okay, it is safe to use those school science laboratories because it does not selectively grow pathogenic bacteria. So non-pathogenic bacteria, they are considered to be non-fastidious. Okay, especially yung mga microbiota or normal flora. So aseptic technique used in the lab, so refers to procedure that is performed under sterile conditions. Mga sterile conditions, free of pathogens, free of microorganisms. Okay, this includes medical laboratory techniques which deals with culture and human cells and tissue transplantation. Okay, so what is the aim of aseptic? To prevent access of microbes or microorganisms during the preparation and testing. So usually when you do the culture medium, so usually in aseptic technique, kailangan naka-gloves naman kayo. Okay, naka-face mask, and then at the same time naka-lab gown, and all the glasswares that are needed, especially petri dishes, undergoes the sterilization process. So gina sterilize using the moist heat process, okay, or autoclaving. General principles of aseptic, so disinfect the working area. So if this is your working area, like in the laboratory, later on if we have a face-to-face, so you have to disinfect the area to prevent possible contaminations. Next, flame in inoculating tube. So flaming, so before and after the transfer of bacteria, example, you get the bacteria from the sample, so you have to do the flaming process, usually red heat ang tawag doon, okay, to prevent contamination again. Next, flame the opening of the glass containers. This is very important, before removing the bacteria from them, and again after the bacteria had been removed. So usually if this is the source of your specimen, usually this is known to be the BUTT, or BUTT, that is a vat form of the culture medium. Usually before you get a sample of bacteria, usually have to pass it in the flame, okay, to avoid contamination again. Next, do not lay the cap of the containers of bacteria on the bench top while bacteria are removed from the transfer to the container. So hindi mo pwede sandin na naibutang ang mga covering. Okay, example sa petri dish, may cover yan eh. So if you get the cover, don't place it anywhere. So kailangan sterilize mo siya muna by flaming, and then you place it in the or you place it in the sterile area. Okay, after na non-sterilize mo siya in flaming process. Next, so work quickly, efficiently to minimize time falls to exposed environment. So you work as fast as you can, kasi most of bacteria are considered to be airborne, so they can able to contaminate once they open the container, or you open the petri dish wherein there is a culture or the agar. So usually exposed na yan kaagad, so you have to do your work very quickly. Okay, next, so aseptic technique in flaming, this one. So if you have the inoculating tube, you pass it to the flame. So usually tawag doon red heat. Then with free hand, raise the lid of the petri dish just enough to access the colony. So parang angle ng 45 degrees lang siya. Okay, and then you dip. So look for the area wherein there is a growth or bacteria, I mean. Then after flaming, the mouth of the what is called sterile stand strict in its surface. So pwede kayo kung gusto mo, coming from this one culture, gusto mo ilagay yun sa ginatawag natin na culture bath. So you have to dip this one using the streaking method. So pwede kayo magkaroon ng zigzag method dyan. Okay, flame the mouth of the tube again, and then flame again the inoculating tube. So what is the purpose of flaming? Of course, to avoid contamination, kasi once you open this one glass, this is exposed already in the air. So there is what we call possibility, I mean, of a bacteria that are going to contaminate your culture. Okay, so take note of our aseptic technique again. Okay.